Okay, hello everyone, and welcome to Karolinska Institute's workshop in the frame of Preserving the Brain, the Forum on Neurodegenerative Diseases organized by Prada Foundation. Our symposium title is Demyelination and Remyelination in Neurodegeneration from Molecule to Clinical Application. We will really like to thank Karolinska's rector, Professor Ole Petter Ottersen, for having us here today in this beautiful venue at the Nobel Forum, and for agreeing also to say a few words to open the symposium. So please, Professor, the floor is yours.
Thank you, Chiara, Tony, Gonzalo, Frederick. You are responsible for this meeting. Fantastic, because we need this kind of meeting. And why do we need this meeting and this kind of meetings? We need because of the title, Preserving the Brain. What could be a more noble mission in the Nobel Forum than to discuss how we can preserve the brain? And I think it's fascinating to look at the visions that are indirectly driving this meeting. One is the mission of Karolinska Institutet. I will come back to that. But the other vision is the one embedded in the philosophy of Fondazione Prada. And we are very grateful to Fondazione Prada for supporting us. And I know that this is now being broadcast to Italy. And we should be aware of the fact that this meeting is complementary to one exhibition in Milan, one in Venezia, and it has everything to do with the brain. The vision of Fondazione Prada, I was really very, very enthusiastic when I read the mission, because what they say, if I understood it correctly, is that I should take basic research, groundbreaking basic research, all the way to a better treatment for the patients, to the clinic. And in fact, this is a mirror image of our own vision at Karolinska Institutet. And you all know what is our vision. You have to learn it by heart, of course you have. Our vision is to advance knowledge about life and strive for a better health for all. And one of the major difficulties at Karolinska Institutet is to bridge these two parts of the mission, to ensure that, yes, our knowledge about life is efficiently translated into better care for the patients. So the visions of Fondazione Prada and KI's vision, they are very much aligned, and this meeting reflects the two missions in a very appropriate way, I would say. So just a few words about this particular meeting. The first thing I would like to say is that I'm extremely depressed today, because as Christian knows, I myself have I would say, very strong empathy, enthusiasm, sympathy for this topic. I've been working on glial cells myself for quite a number of years, but I'm depressed because of all the duties. There are other duties that prevent me from taking part in this meeting. Had it not been for the other duties, I would have been here for the entire meeting, I tell you. There is one other issue which I think is essential and which we should communicate to our friends in Italy, and that is that the starting point for this meeting is something that is also very close to my heart, and that is the deep understanding of events at the single cell level that our scientists here at Karolinska Institutet has brought to the fore. Single cell technologies, I think one could safely say that KI is famous, world famous, for its research in single cell technologies. It's a long distance from single cells to diseases like MS or other neurodegenerative diseases. But, well, it's a task that we have to handle and to cope with and to address. And one way to address this tremendous gap between single cells and diseases is to have meetings like this. And not least meetings supported by foundations like Fondazione Prada. So with this, I would simply say have a good meeting, rewarding meeting, and I do hope that you will arrive at some new ideas as to how to leverage basic science into the clinic and to better treatment for the patients, because after all, that's why we are here. Our social contract, our contract with society depends on our ability to translate science into better treatments. So it's a noble mission in the Nobel Forum, and I'm sure that you are aware of the fact that there could be no better place for a discussion like this. Thanks for your attention.
Yes, thank you very much, Professor. We really appreciate your time that you could dedicate us today. We know you're very busy, so we really appreciate that. Thank you very much. So now we will start with our fourth scientific presentation by our invited guest speakers, and every presentation will be followed by a discussion on for a Q&A session, and then after the speakers, we will have a panel discussion on perspectives in the field and opportunity for translating experimental results to the clinics. For the Q&A session, you're kindly welcome to ask a question both from the audience and from the Zoom session for those who are attending online through the Q&A tool. And we kindly ask then the speakers to repeat the question here in the microphone so everyone can hear it. So yes, I hope you enjoy all the presentation and the discussion. And please, Gonzalo, if you can come here and introduce our first guest.
Okay, thank you, Chiara. So now it's my pleasure to present the second speaker of this event, Professor Maja Jagodic, who is a professor in neuroinflammation here at Karolinska Institutet. Professor Jagodic is one of the responsibles for the European Consortium Multiple MS, aimed to disentangling phenotypic manifestations of the disease through a multi-omics approach. Professor Jagodic's research group focus more on epigenetics origins and mechanisms of multiple sclerosis. And this is the topic that Professor Jagodic was going to talk. So go ahead.
Thank you.
There was a question. Well, we can take it at the end.
Okay. But how do I see where to point?
So let's see. Yeah.
And this is yes. And
okay. Thank you very much. Thank you for the invitation. It is my pleasure to join this great initiative. And what I would like to discuss today are some perspectives for molecular markers. What I will show and raise are mainly going to be various hypotheses, but I think that
is not sharing.
Okay, is it shown? Yes. But I think that there are promising prospects in this field. So what we are focusing on mainly, and this is not moving. Okay, thank you, is multiple sclerosis. I don't think that this audience needs much introduction. I would just like to stress that there has been really an enormous progress in treating these early inflammatory stages of MS, and there is a lot of research ongoing into understanding the secondary progressive stage. What we are still lacking are robust molecular biomarkers for the disease stage, state, response to treatment, or any other disease-related processes and factors. So my group is in that context studying epigenetics. The reason why we do that is because this layer, the epigenetic layer, is taking the instructions from the genetic variation from different exposures, as well as from the disease itself and different treatments that people are exposed to. So by looking at this level, in particular changes in DNA methylation and non-coding RNAs, We are trying to understand the disease mechanisms, to find classifiers and biomarkers, and hopefully come also to potential novel therapeutic strategies. But what I will talk about today is some insight from looking at changes in DNA methylation and particularly small non-coding RNAs at the different compartments in blood and plasma, in the cerebrospinal fluid and cerebrospinal fluid cells, as well as in the tissue. And just raise some ideas and hypotheses that are coming from this data. A while ago, we started to study genome-wide methylation in 4 different immune cell types from blood, CD4, CD8, monocytes, and B cells, and we found also some shared changes that could classify patients that have MS in general and those that are in the secondary progressive state. And what we could see from this data is that when it comes to MS in general, these methylation-associated changes were implicating dysregulation of the adaptive immunity, T cell, B cell receptor signaling, T cell activation, so nothing really unexpected. And there was a lot of colocalization with the MS risk genes. What was interesting is, what did we pick up when we look at the secondary progressive stage, the pathways? Here, the innate immunity myeloid compartment was much more implicated, processes such as phagocytosis. And then we also saw some neuronal and neurodegenerative disease genes. And just remember, these are the cells from blood that we pulled out and analyzed, the immune cells. So we replicated this neuronal signature in an independent cohort, but we also started to think, are there any shared changes? Because this has been shown in neuropsychiatric and developmental diseases that you can find this epigenetic signature both in blood and in brain. So we overlapped some of these changes in the immune cells in secondary progressive patients with the changes that were observed in the tissue, in the neuronal nuclei of MS patients that we did, as well as other studies that look at hippocampi or white matter of the brain. And we could see that for those immune changes that we observe in secondary progression, there was a lot of functional overlap with the neuronal functions. So why do we see this neuronal signature in blood? Of course, it could be that something that we call typical CNS genes are also having immune functions. There is, of course, an increasing evidence of this happening. For example, the glucocorticoid receptor is very important for activated T cells. The glutamatergic receptor. But it could be also the exposures, because we know that different exposures and environmental factors are imprinting molecular changes across different tissues. Could also be aging processes, could be genetics, and so on. So I will give you a couple of examples that might indicate that some of this could be true and give us a tool to address and molecularly tackle these processes and learn more about them. So first is exposure that I will talk about, smoking in particular. Why? Because we know that smoking associates not only with an increased risk of developing multiple sclerosis, but also with the risk of having a more severe disease and more progressive disease. So we looked, what are the changes that smoking is inducing in the methylome? First, of the cells that are most exposed, bulb cells from the bronchoalveolar lavage. And we see if we associate these changes in the bulb cells that are predominantly macrophages with different functions, they're usual suspects for how the smoking would impact inflammation, because it affects the cellular migration, adhesion, trafficking, different inflammatory responses. This is one of the hypotheses how smoking can trigger a disease such as MS. But then we also observed this nervous system and neurological disease pathways, and they were particularly pronounced in MS patients compared to controls. There were very few significantly enriched functions in healthy individuals, but they were there in MS patients. Now, smoking is known to be associated with better brain performance and functions, but why do we see this much stronger in MS? We have previously observed that there is an interaction on the molecular level between the smoking and the disease itself. This is looking at the peripheral blood and demethylation that occurs at one site, but we have seen this on many sites, in the response to the amount of smoking as a pack years. So we see that this is the demethylation that occurs with the amount of smoking in healthy individuals, and here is the demethylation that occurs in MS. and correcting for confounders, this is a significant difference. So there is some sort of an interaction between the disease processes and this environmental exposure that makes the molecular changes even stronger. So what we are potentially picking up in different tissues could be a response to some exposure that is then in the brain and in individuals affected with brain diseases leading to progression. Another factor that might be related to that is aging, because we know that age is the most significantly associated with the conversion to secondary progressive MS. So we exploited the possibility of estimating different types of biological aging through different epigenetic clocks, looking at the DNA methylation. And in blood, we could see that MS patients are exerting significantly accelerated pheno-age. Phenotypic age is a specific biological and epigenetic clock that is a very good proxy for all-cause mortality, morbidity, and often actually connected to inflammation, to the chronic inflammation that starts over the years. So it could be that MS patients have increased biological aging because of the chronic inflammation. Then we also looked at cells in the brain using the cortical clock, which is also a biological and epigenetic clock that is more suitable for the brain tissue. And we could see, especially in the glia, after correcting for confounders, that there is an increased aging of the glia cells in MS people compared to non-MS controls. So it could be also the process of aging potentially accelerated by the chronic inflammation that is imprinting these different marks across the tissues, but has a functional effect in the tissue that is implicated in this condition. Then it could be also genetics. It could be that we have a genetic control of epigenetic marks and gene expression that is often observed across different tissues. But again, it's important where the functionality happens. And we have potentially one such example. So we've observed that there is a differentially methylated region on a number of CpGs where methylation was higher in the primary progressive MS patients compared to other groups and also healthy controls. From this pattern of methylation, we could see instantly that it's genetically driven because you see these different categories of the levels that is typical for a genetic regulation. But the locus itself was extremely interesting. So this is the locus where there was the largest copy number expansion going to from non-human primates to humans. And these copy numbers are associated with the brain size, with motor, cognitive functions, with neurodevelopmental, neuropsychiatric diseases. So it was extremely interesting to look at it. So we started to speculate that maybe we have a genetic predisposition for primary progressive disease or maybe more severe disease that is then imprinting these changes in the methylation across different tissues. And in this cohort, we did observe some evidence for association, although this is quite weak, but it's difficult to find large cohorts with primary progressive MS. But we went on further to see whether there is any evidence of this type of a cross-tissue regulation. And we observed that these genetic variants that associate with PPMS in two different cohorts, they also associate with an increased methylation levels in this locus in blood. And then we looked at the brain XQTL data from Phil de Jager, and we observed that the same variants are associating with increased methylation or changes in methylation in the brain tissue, and also with changes in expression of genes that are residing within this locus. And now to confirm that methylation here indeed regulates the expression of the genes, we use this neuronal-like cell line where we specifically with the DAD Cas9 and fusion with the TET demethylated the locus. Removing the methylation at this locus led to increased expression of these two different genes, CHD1L and PKRB2. So they are controlled by methylation in this locus, their expression. And then we used data from Patricia Casacha, RNA sequencing data from post-mortem brains. and perform network analysis. And we found 2 networks that associated specifically with primary progressive MS. And one of them had this CHD1L as a central gene in the network. This is a helicase that is important for DNA replication and repair. And the genes that are connecting with it associated with neuronal functions and neurodegenerative diseases and neurological diseases. So this was really interesting. Then we tried to replicate these findings in another context. We could not really see this network in healthy individuals and in the data from healthy brains. But when we went to single nucleus RNA sequencing data from Lucas, and specifically this COX-2 expressing excitatory neurons that were shown to be affected in MS brains, we could replicate the most immediate interactors of this network. So it seems to be something specifically established in the context of a disease in the neurons that likely associates with neurodegeneration, potentially genetically driven. There are also indications that there are both genetic and epigenetic implications for microglia being important in MS. Here is our data from methylation analysis that is implicating different functions in MS brains. And here is one of the biggest GWAS analysis that we did, where a certain number of the hits are also going towards the microglia and implicating microglial genes. And we found some of the processes that we were interested for a very long time, and that is autophagy and phagocytosis implicated here. If you remember, I've also mentioned that we've seen evidence from the immune cells of phagocytosis and myeloid cells being important for the progressive disease. So in this context, we knocked out one of the main autophagy genes, specifically from microglia, and then we could see that these animals never recover. So they remain in this constantly progressive stage. And we could show in very many different ways that they have impaired degradation of the myelin. This is changing and shaping up the microglia into a completely different phenotype. There is inability to clear myelin debris, which is affecting proliferation of oligodendrocyte progenitors, and also the ability to remyelinate when you cannot remove the debris after the initial insult. What was interesting is that transcriptome of this microglia is extremely similar to transcriptome of aged microglia. And aged animals also cannot recover. So we were wondering, can we do something to boost this process so that the animals can actually get better? If you focus here just on the black lines, this one here is aged mice that do not recover, similar to microglia ATG7 knockouts. If we give them a trecalose, which is a simple disaccharide that boosts autophagy and phagocytosis and myelin clearance, then they can. So these data are suggesting also that likely the inability of microglia to clear myelin debris or any type of a debris during this type of insult that is exacerbated by aging is responsible for progression of the disease. And there is a potential therapeutic avenue here if we can boost these processes. So to sum up, methemoglobin immune cells support dysregulation of adaptive immunity, dysregulation of myeloid cells and phagocytosis in disease progression. And what is interesting is that maybe this methylome can give us potential markers from which we can learn more about other disease-associated processes and also the disease development and progression. Maybe we can learn more about the exposures or just detect different exposures and also genetic predisposition. And it's likely that these processes are going to determine the vulnerability of the target tissue and the ability to repair after the initial insult. But this is really in its infancy. We need to learn how to associate these different molecular changes with this type of processes and how to disentangle them. And it's not always convenient to measure DNA methylation genome-wide. So do we have a more accessible markers? We do, and I will just show a few slides, and that is non-coding RNAs. So there is an exploding world of non-coding RNAs that have different functions. And if we just focus here on small ones, we know that very many of them are important for basic functions such as protein synthesis, spliceosomes and alternative splicing, inhibition of protein synthesis and RNA degradation. So we got interested whether we can get the biomarker potential and learn something about the disease from these small RNAs. So we adopted a single cell pipeline, small-seq to low input samples from clinical cohorts. And we profiled 48 individuals with the matching compartments, PBMCs, plasma CSF cells, and cell-free CSF. And we could find a lot of different classes of small RNAs, also that they are different in the periphery compared to the central nervous system compartment. And they were present in fragments, but also in the longer forms. And what is interesting is that depending on their size, they can do different functions. And many of these functions are still not even known. What was really shocking is that there was this opposing pattern at the periphery compared to the CNS compartment. If you look at this dysregulated small RNAs, you see that in the PBMCs, they're downregulated. Very many of the same ones are upregulated in the CNS compartment. This is really important for looking at their functional consequences. And just zooming on tRNAs, we see that they're increased in the CSF cells. They're very often decreased in the PBMCs. Very many of them we can also detect in the body fluids, which is interesting. Their fragments have a different distribution across the compartments. It's very interesting that in the CSF cells, we see these 5' halves, and they correlate with the processes and protein biomarkers of processes that they can be involved with. So then the question was, can they have a biomarker potential? So what we did here is an unbiased analysis across all of these different small RNAs and also different pipelines to see whether we can distinguish cases from controls. So here we do not really say whether somebody has a disease or is a control. We were just looking at the components that are differing the most in these subjects. And then according to the first component, we could actually separate with area under the curve of 82 or 0.82, which is quite good, RRMS from all controls together. Projecting also SPMS, we see that they land somewhere in the middle. And we also see what kind of small RNAs and pipelines are contributing the most. So we see that the small RNAs and especially tRNAs in the cell-free CSF and CSF cells and plasma are contributing to this separation. So we have alterations in several classes of small non-coding RNAs, not only microRNAs that have been typically studied. There are prominent changes, particularly in the CNS compartment. There are opposing changes in the periphery versus the CNS, so we have to be extremely careful. These different functions are implicated, of course, and there is a biomarker potential for small RNAs, and we can measure them in a fairly simple way across very many samples. So there is a potential future for these type of markers, but it requires a lot of additional work and replication. And with that, I would like to thank my group, a lot of collaborators and funding bodies, and also you for your attention.
Thank you, Maja. Questions from the audience? Yeah.
I have a question.
Frederick?
Thank you, Maja. So you show the phenoage in immune cells is quicker in MS patients, and then in the brain cells. But is that all body tissues, or is it just the immune cells and the CNS cell?
We haven't actually looked at other. Oh, should I repeat? There is increased phenoage in immune cells and increased cortical clock in brain cells. And Fredrik is asking, is there increased aging in other cells? So we have not looked systematically at it because we don't have methylation data in many other tissues. We have looked at cells from bronchial lavage, and there are some changes there that are still a bit difficult to reconcile. Lara is working on that. So I think we're most likely going to pick up changes, at least those that are related to inflammation in many tissues, because if they are driven by inflammation, they will affect different tissues. But one also has to consider what type of clocks are suitable to be used in different tissues, because they've been trained on different data types.
I have a question about the 1-221 locus. So you see differential methylation specifically in PPMS patients, so primary progressive, but you don't see the same pattern in secondary progressive. So could you imagine like there are different risks for the degeneration and progression in these two phenotypes?
I mean, we see that this increased methylation in PPMS is there because of increased frequency of this variance, specifically in PPMS. Now, the cohorts are really too small to be able to say that conclusively. So, but also the network analysis data from these patients indicate that there might be potentially some changes between PPMS and SPMS, although I cannot really say. I would say that most likely a lot of mechanisms are shared, but potentially there could be some differences.
Okay. What?
So do you think it would be feasible to slow the aging process by modifying your findings? I was really interested in the coastal microglia, the mature and the aging, and at least in our hands or previous work, then it's the astrocytes which are really important. You put young astrocytes with old microglia and they're really young. So I wondered, do you see potential with the microRNAs or RNAs to actually
with different types of molecules? Yeah, I mean,
the aging process.
There is a question from Bob Harris whether we could revert or slow down the aging of these different cells and have beneficial. Well, we're of course, I mean, a lot of different groups in different contexts are working on that, and there are compounds that can do it. There also, autophagy itself has been implicated in rejuvenation and different interventions that can boost this process. So one can probably fight it to a degree with certain type of lifestyle changes. But at the same time, I think we would also need to control the inflammation and other processes to get a really true clinical impact or something like that. We cannot really work only on one arm. And then, of course, there are a lot of people who are considering whether epigenetic modifications can revert these aging processes, but we don't really know if these changes in epigenome are just a marker or if they have any functional roles.
Okay, I have a very provocative question to you, because I'm very interested with the tRNA fragments, and there were some studies some years ago from Oliver Rando suggesting that they could be involved in transgeneration inheritance. It's very controversial, but I'm just wondering, because you are seeing these different patterns, if you could see that within the body, these tRNA fragments could also be communication between different, like you say, you see changes in the lung and then in the brain. Could it be that they are circulating and they can?
I think that they could potentially, especially because there are lots of different functions that have been assigned to fragments. And it seems like every now and then, very frequently, we're hearing about new functions in the protein synthesis, in changing the chromatin, in acting like microRNAs, in regulating stress responses. So this field is really exploding. And most of them are, and that's probably a reason why we detect them, are in microvesicles. So as much as microvesicles are included in communication, I would say there is a potential for these small RNAs to be included in communication between different parts of the body.
Okay, with that, thank you very much.
Thank you.
So last topic for today, something that lies close to my heart being an MS clinician. Although I'm really waiting for the moment when I get this in my kind of clinical hands. So I'm happy to introduce Tobias Granberg, who's leading the MRI effort here at Karolinska, and you'll talk about in vivo imaging of and demyelination. Please, Tobias.
Thank you, Frederick. And thank you also for the kind invitation to give this talk. I'm a radiologist, so I come with a slightly different perspective, but hopefully I'll be able to show you a lot of pretty pictures, but also some images that's very close to clinics or in clinics as well. So let's see. All right. So before I start this talk, I just wanted to let you guys know that I have no conflicts of interest with regards to this talk. I will be showing you some imaging techniques that are actually commercialized products now, but I have no financial conflicts of interest. And I think it's this saying, seeing is believing. I think it cannot be over-exaggerated how true that is. And I would like to stretch this a little bit further and saying seeing and quantifying is believing. And when it comes to imaging and its role in MS, it has had a very important role, not least in diagnosing patients correctly as early as possible and also offering them early treatment. Imaging has also played an important part in developing these different treatments that we have available today. The real breakthrough when it comes to finding treatments for MS actually came when we started applying imaging to this spectrum of patients with the diagnosis of MS, which is a spectrum from a benign, rather benign disease to some patient having very disabling disease. And when starting using imaging both to screen patients, to identify the patients that were potentially the most vulnerable and the patients that could have the best benefits of treatment, and then using also imaging as outcome measures. And I will show you some examples of how we're doing that today as well. And it's always nice to get some sort of perspective historically. And the very first image of a brain MRI scan in MS was performed in Queen Square in London in 1981. And since then, we've come a far way. This is an example of a modern 7 Tesla image. And you can, of course, appreciate the difference in terms of spatial resolution, the soft tissue contrast, but more importantly, you can see some very interesting pathologic features. And you can see here with the yellow arrow, you can see this is actually a cortical lesion, not white matter disease, but actually gray matter disease. And you can see that you have these small veins in there. And we know that MS is a heterogeneous disease. So this makes it very interesting to try to explore this a little bit further. And then again, why does imaging matter? Well, we are very much familiar in the clinical context of stroke, of the concept of tiny brain. We do our very best to have the patient in the CT scanner as early as possible for interventions to delay the damage to the brain of the an ischemic stroke, for example. But the same is true for MS as well. It's just that these processes take longer time. So finding the patients, diagnosing them as early as possible, and then treating them correctly is essential to hinder the accumulation of disability. So how does this look in practice? I usually pull out this graph, and every time I do a new lecture, I usually update this graph. And every time I feel like, oh, this is not going to get any better. But every time I'm surprised that we continue to see this decrease between the symptom onset to the diagnosis of patients. And there's a lot of factors going into this, of course. I mean, there's patient delay, doctor's delay, increased in that awareness of the disease. But most importantly, this is about getting the diagnosis. And to get the diagnosis, you need to apply the diagnostic criteria of MS. And this has changed over time. And actually, MRI was introduced in 1996 in the diagnostic criteria. And since then, we've had several revisions which strengthens the role of imaging. And this, I believe, has helped a lot in finding patients that we can treat as early as possible. So before we start looking at images, it's important to understand what we're actually looking for. We know that MS is a pervenous disease, and we also know where the veins are in the brain, of course. And it's typically these smaller veins called venules that are affected by MS. And if you imagine that you have an inflammation starting around the vessel, which is a tubular, typically a tubular shape, what will happen if you have a migration of inflammatory cells around this tubular shape? You'll actually get these ovoid shapes, right? So that helps us when we're looking at the brain images and trying to figure out is this MS or not. If we see white matter changes in the brain and you can see that they're ovoid, it actually speaks in favor of the diagnosis of MS. But what happens then if you are close to the ventricles and the CEST barrier? Well, you have the ependym close to the ventricles, and since there is a barrier there, what will actually happen is that you get this almost pyramidal triangular shape, which is also very typical of MS. But MS also affects the U fibers of the brain. That is the small connective fibers in between different cortical areas. And if you have an affection of those fibers, you will get these U-shaped lesions. And MS also affects the gray matter, and I'll show you some examples on how we can visualize that in vivo. So this is an example of what modern MS imaging looks like. This is a side view of the brain. It's a 3D 1 millimeter cubic resolution scan. And this was actually taken in 2015. So since then, it's actually gotten a lot better, I would say, in terms of image quality. But in 2017, we agreed as a community in Sweden that all MS patients would be imaged with 3D imaging, high resolution 3D imaging, which helps us to do some of the applications that I will show you later on, to not look only for small lesions, but also look at the atrophy patterns of these patients, to look at what is probably closely related to progression. So something that's a little bit of a buzzword right now is precision medicine. But and actually one of the initiatives between KI and the Karolinska University Hospital is precision medicine in MS. It's actually two selected pathologies. There's lung cancer and then there's MS. And one of the reasons why this is selected is just because of the plethora of different treatments that you can see in MS. This is just an overview showing you the amount of treatments available and how patients go through different treatments across through their disease progress and how they switch between treatments. So understanding which treatments work the best for the right patient and person is super important. So how can we then try to quantify the disease so that it's easier to evaluate the treatment effects, especially in real life contexts? Well, this is an example from my daily practice. This is an axial size of the brain of a patient with MS. And now we actually, typically what happens if you do an MRI scan is that you have a radiologist or a neuroradiologist assessing the images, and then we subjectively assess how these images look, and we try to put that into some sort of plain text. Since 2015, we're doing this as a standardized reporting, which helps the doctors put the sort of summarized view into the MS registry. But more importantly, with these 3D images, what we can do is actually segment the lesions, which means that we actually highlight them in a picture like this. So this is something that can be done automatically. It's actually something that we have activated in the clinical practice as part of this precision medicine initiative. Here, it's an AI tool where the computer is essentially trained to find MS lesions, and it helps us pick them out. And naturally, I can see these lesions as well. But more importantly, when I get the help of painting them in the picture, it also helps me quantify them. So now I can actually not only say that there's a lot of lesions or few lesions or 23 lesions, but I can actually say what the amount is in milliliters. And this makes it much more quantitative, and it makes it possible to actually use it as some sort of outcome measure and compare between groups. More importantly, we've been talking a little bit about the progressive phase and the neurodegeneration. And brain MRI is actually fantastic in looking at accumulated damage across time or injury. And this same tool also segments the brain. It tries to find these different anatomical structures and then quantify how large are they. And then we can actually look at atrophy patterns. We can see if there's different subtypes of neurodegeneration in these patients. And it's very likely, as we've seen before, with finding NMOSD as being actually another disease than MS, that these patterns might tell us that we have different subcohorts of patients within the spectrum that behave differently. And when it comes to the injury to the gray matter, this is something that's been known since the early anatomists did the first brain sections in MS patients. However, the knowledge was lost a little bit because part of it was that when you wanted to look at the myelin and demyelination and the scars, you often do these luxopath blue stains. The protocols typically say that you should wash out a lot of blue until the cortex becomes more transparent because you have less myelin there. But that makes it really hard to see if you have lesions in the cortex. And when MRI came into the stage, and we typically couldn't see the cortical lesions, we sort of forgot about them a little bit. But now we're seeing a renaissance where we're increasingly focusing not only on the white matter, but also of the gray matter in these patients. And these are just some examples from our clinical practice. And you can see here with arrows that sometimes, even with conventional MRI scans, you can actually pick out these cortical lesions. And we know that when we see these, they are just the tip of the iceberg, but they are significant because it means that these patients not only have a worse prognosis in terms of their clinical outcome, more importantly, it's associated with cognitive disability, which might be very hard to pick up in a clinical context when you're just talking to the patient. And in this context as well, it's also interesting to know that we don't really know if we're treating these lesions or not when we're administering these disease-modifying therapies. And part of not knowing is because it's really hard to visualize them. So what can we do to try and do better? Well, one thing we can do is increase the field strength. So I'll show you some examples of work that we've performed in collaboration with our with research groups in Boston. And here with the arrows, you can actually see these cortical lesions much more clearly because you have a sub-millimeter resolution. These are actually 330 micron in-plane resolution scans. So you can get almost microscopic resolution in vivo in these patients. Something that's interesting in this picture is if you look at the top arrow, you can actually see that you have this lesion that looks almost like a mouth almost, and it's around something black, and the black thing is actually a vein. So even the cortical lesions seem to be pervenous. And interestingly enough, if you look at early MS patients and you scan them with 70, it turns out that most of these patients actually have cortical lesions. It's just that we can't see them on conventional MRI. And if you follow these patients over time with 70, what you'll see is that you actually get more lesions in the cortex across time than in the white matter. And we know that these lesions are actually more strongly correlated to disability than the white matter lesions. So perhaps we've been looking a little bit on the wrong side of the coin when we're talking about preventing disability in MS. Interestingly enough, when you have these higher field strength scans, you also get much more sensitive to iron. And we know that myelin contains a lot of iron in its myelin complex. When you get the degradation of myelin, what happens is that you release a lot of iron, and then you will have these phagocytic cells popping up, and they'll engulf some of this iron. but then they will stay on site, which means that we can actually see this in vivo. And you can see here that these linear lines, they're actually small veins, but along them, you can see that there are depositions of iron. And more importantly, you can see that there's actually a ring around the lesions that are iron as well. And if you find these iron rim lesions in patients, these actually turn out, if you do histopathology, turn out to be chronic active lesions. So we believe that these are lesions that we can't really see by naked eye that they're expanding. If you follow them across 3.5 years, perhaps you can say that you see a little bit of a progression, right? But it's really hard to pick up. But these lesions have chronic activity, and we don't know if we're treating it, and whether there are treatments that we have now that some treatments might be useful or not. So currently we're focusing a lot on these lesions because they also seem to be correlated to progression in MS. So you have patients who have no new relapses, they have no new focal MRI lesions, no contrast enhancing lesions, but they still get worse. And the question is why? And then if you look at those patients, what you'll see, a lot of them actually have a lot of cortical lesions. The other thing you'll see is that they have these types of lesions, which means that you have some sort of chronic activity ongoing. More importantly, also with the topic of this talk, we can actually also look at myelin in vivo with MRI. So Christian showed earlier, when you have this myelin sheath around the axons, that you have, it's essentially like a cinnamon roll, right, around the axons. And in between those cinnamon roll layers, you'll have some smet in Swedish, goo, the tasty stuff in the cinnamon roll. And you have the same thing happening with the myelin complex, but you actually get a little bit of water in there that's trapped. And interestingly enough, that trapped water will have completely different diffusion properties than anything else. And that is something that we can measure and visualize. So something that we've been working on, and especially Russell, who did his PhD on this, is looking at trying to visualize myelin in vivo using MRI. And with MRI, you can become more and more quantitative, you get a high resolution, but in the end, and you can do colorful pictures as well. But in the end, if it's not validated, it doesn't really mean anything. So what we've done with this technique is both show that it's a very robust technique. It has a higher reproducibility and repeatability, but more importantly, it actually shows the myelin and it's correlated to the myelin content. And then if you start looking at using these tools to look at the MS spectrum as a whole, you can find some interesting things. So on the top left there, you see the control, and how the more yellow it is in the picture, the more myelin you have in the voxels in the brain. And if you look at the relapsing, remitting MS patients, you can see that obviously they have a lot of focal lesions on the flare over here. And then if you look at the myelin quantification, you can see that you have a loss of myelin. But interestingly enough, where we on the MRI, it sort of looks the same white everywhere. But if you look at the myelin map, you can actually see that there are differences within the lesions with a varying degree of demyelination. And if you go from RMS to SPMS, you can see, well, the focal lesions have increased to some extent, but perhaps not that drastically. But more importantly, you can see that the whole brain is a little bit less yellow. So with these techniques, we can not only see focal lesions, but we can also see the diffuse demyelination happening in the brain in the normal appearing tissues. And interestingly enough, if you look at the PPMS patients, they seem to behave differently when it comes to demyelination. So one can wonder, is it perhaps a different disease or disease entity? Or is it perhaps that they have the same type of disease, but they just have a better remyelinating potential? We don't know. Interesting enough, we can also, this is the same technique where the more yellow it is, the higher myelin content you have. And you can start applying these concepts also to other diseases like the NMO spectrum disorder patients, the ALS patients, and even patients with COVID-19 and cognitive impairment. So currently we've shifted from just seeing lesions to actually being able to quantify them and becoming increasingly more tissue specific. And I think that goes along the lines of what we're hoping to do in collaboration with other researchers here at KI. And I just wanted to leave you lastly with this image where we're trying to develop these myelin imaging techniques to also work in 3D with high resolution. So this is something that we're currently working on. So with that, I would like to thank all the participants of our studies, our funders, and of course the research team and our collaborators. And lastly, I would like to thank all of you for your attention. Thank you.
Thank you, Tobias. And any questions? Are you curious about the myelin content called Tobias?
Bob?
It's fantastic and it's wonderful that if you're an MS patient in Sweden. But how do you see the future that this will be used in clinical practice the world over?
So the question is, how relatable are these techniques to globally? And actually, we've developed this technique together with collaborators in Linköping, and it's actually now available. If you buy a new MRI scanner worldwide, you will get this technique on your scanner. And it's actually available on all the major MRI vendors. So it is a technique that's out there today, and it's CE marked as well for clinical use.
MRI machines are quite expensive, so clinically around don't have the possibility.
Sure.
See that it should be, because the earlier is to see the patient, right?
Well, currently we don't know how actionable the data is, right? Because we haven't even had the time to ourselves look at the data, and that's something we're currently looking at. But if these imaging techniques actually give us, which we believe, more data and earlier data, more specific data, then obviously we want it to be as accessible as possible, and that's also why we're not profiting from it. So, yeah. Yes, please.
Finding correlation between the site of lesion in the cortex and the type of treatment.
So the question was whether there's a correlation between treatment and the localization of lesions in the white matter versus the gray matter. So actually, and also interestingly, in terms of the CSF barrier, which is one of my favorite topics, we actually published a study recently in Brain, also with Russell as the first author, looking at the difference of gray matter versus white matter pathology, both in the brain and the spinal cord with 7 Tesla. And it seems like most patients actually have gray matter injury, both in the spinal cord and in the brain, and it's correlated. So if you have a higher degree of gray matter injury in the brain, it will correlate with the spinal cord injury as well. In terms of treatments, we still don't really know, because typically these 7 Tesla studies are with quite few patients. So the scanner that we worked with was a non-CE marked product, and we scanned, when I was a postdoc there, we probably scanned like maybe 30 patients in a year, and we had a cohort of like 100 patients. But if we would have this in a clinical context, the numbers would be completely different, because we do like 3,000 MRI scans in MS patients per year. Here at Karolinska, we have about 27 MRI scanners, and we do a lot of brain MRI. So yeah, with the right tool in the right place, I think it could be substantial. Anna?
Yeah, so what do you think is the killing mechanism in the cortical lesions compared to the white matter? Is it still the myelin that's being attacked by the immune system, or is there something else going on?
So the question is there a difference in the pathological mechanisms of cortical lesions versus white matter lesions? Based on imaging, we can't really say for sure, of course, but it seems to be very similar processes, because they both, the lesions appear close to the veins in both compartments. And more importantly, we've also done some PET MRI studies, and you can see, speaking about microglia, that you get microglial activation in both the white matter and the cortex. But on the other hand, microglia is sort of like the fireman, right? Just showing up where there's inflammation, demyelination. So they might be a little bit of a innocent bystander. You have different subtypes of microglia, etc. So it's still a little bit earlier to say what the exact mechanisms are, but I would expect them to be similar based on the imaging findings. Gonzalo.
What about remyelination? So if you do, how can you detect, or is it, I mean, with the advances in technology, can you really focus on there and really see progression?
So the burning question is, can you see remyelination, right? And the answer is yes. So we have actually done with the Professor Fredrik Pilas, the PI here in Stockholm. We've done a study where we've had patients who have been treated with rituximab, anti-CD20, highly effective anti-inflammatory treatment. But the patient without relapses, without any MRI activity, is still progressing and becoming worse. For those patients, they were offered inclusion in a clinical trial, a phase 2 study, where they were randomized between having rituximab plus placebo or rituximab plus an autoantibody that is supposed to essentially reduce the levels of a human endogenous virus, retrovirus type W, that's been overrepresented histopathologically in MS lesions. And then we applied these techniques to see, okay, what is the myelin content before you start treatment, and then see across time what happens. And most importantly, we've been, thanks to Elisabeth, who's done a lot of hard work, segmenting these lesions with really high accuracy and looking within the lesions, can you see a signal of remyelination? And I think the results are promising, but I don't know how much I can comment on it. That's up to you, Fredrik.
Just as a comment, I think that's a really important question, and we have on the clinical side, when Gonzalo and all people working on the preclinical side, at least we can say we have some measures already in place to do trials. So that's an important take-home message.
Yep. Sebastian.
Thank you for the presentation. My question is, this is all in the context of MS, so we associate lesions with the demyelination. There are other neurodegenerative diseases where lesions occur, but they're not really caused with the demyelination. In your presentation, in your interpretation, they are the same thing. But are there different ways to image a lesion that wasn't specifically associated with demyelination? So lesion versus demyelination, can we distinguish that?
Yes, I think that's specifically what this technique is all about. So in many, regardless of what happens in the brain in terms of different processes, and I'm talking everything from traumatic brain injury to inflammation or ALS, the type of Wallerian degeneration without like primary demyelination, they will all present with an increased amount of water in the tissues, pathological fluids, and a decreased signal of fat and lipids. So they will all look bright on these FLAIR images to different degree. And the whole idea with this technique is to try to tease out what of this signal is actually myelin related. So I would expect, if we go back and look at the ALS patients, that if you look at the corticospinal tracts, you can actually see, if I have the cursor, see. Anywho, in the ALS patients, you can actually see in the middle, there's like 2 dots, one on each side. That's the corticospinal tracts, and those go through Wallerian degeneration. So this means that they will be brighter on the MRIs, conventional MRIs, but more importantly, you can actually see that there's some demyelination happening in that process, but that's expected with Wallerian degeneration. But the rest of the brain is rather yellow, right? It looks not like the healthy volunteer perhaps, but close to it compared to the MS patients. So here you can say that there's definitely something different between these pathologies in terms of their mechanisms.
Thank you.
Thanks.
Okay, then we have a few questions from the net, I think. So we'll
yes.
ask the.
I have a question from the audience, from Enrica Boda in Turin, Italy, to Dr. Falcao. So if you come here, please at the podium.
Well, thanks to you.
So great talk. Did you try to transplant choroid plexus from PLP-DTA mice to the control bicycles?
No, we did not do that, but that's something that, yeah, it's a good suggestion. We will keep it in mind, but the answer is no. Sorry.
And then there is another question from the Zoom audience for Christian Gorik. And is asking about how is the response of type B parasites with myelin B?
We call type B parasite everything, I mean, all other non-fibrotic responding parasites, and we haven't seen, let's say, none of them leaving the vessel. So that's definitely, I mean, that's why we distinguish them more like in terms of whether they respond to fibrosis or not, and that's that capacity is completely in the type A population. So there is rather, like say, in the initial phase of the wound healing, there's angiogenesis going on, and in that phase also type B parasites proliferate because with the sprouting vessels.
Thank you. So thank you very much. I would like to invite all the speakers and the hosts to the panel discussion. And now just a few seconds of patience while we move the camera for the Zoom and the YouTube audience. Thank you.
To prevent inflammation and new injury once we diagnose the disease. But the big problem, the big challenge is to do something about existing disability and the progressive disease course. And here we have to move towards regenerative medicine. And I think here myelin is at the core. And the complexity is staggering. We heard from some of the talks that hundreds and maybe thousands of samples will be needed to dissect this multifactorial, and then we have to have the imaging too on all these patients to really quantify disability, and then preferably also choroid plexus biopsies, perhaps from as many as possible. And of course, we'll never come to that stage. But there is a problem of integrating knowledge and how to now collect very large and well-characterized materials and then combine it with the techniques we have at our hands right now. So I open the.
How can we combine what we are doing in basic science to like what you are doing at the clinical level?
Spontaneously, I mean, come up with some ideas to your talk, but one thing that we've been looking at a lot is this inside-out hypothesis about the demyelination starting around the ventricles and then sort of moving up, which is actually what we see across time when we look at longitudinal MRI. So it would be super interesting to look at how this the interaction with the choroid plexus and how that interacts with the imaging over time, because I think it's very nice with histopathological specimens, etc., but in the end they will not be representative for the whole spectrum, right? And they will not give you any dynamic information at all. So if you want to look at the processes over time, you need to use some sort of imaging technique.
If I can add something to this, I told you that we cannot indeed correct the biopsy of the liver. The tissue we have is postmortem, but we can look by imaging, right, by MRI.
Yeah.
And there has been at least one study that's been done to look at cortex. I don't think they looked through time, but it would also be interesting. But they were actually kind of looking. The conclusion, I don't know how to analyze MRI, but the conclusion was that they actually seen that the cortex were more inflamed in the veins. I don't know, how can you see?
Yeah, I mean, you can use PET for that. It's actually, I think we're looking at in Boston. I don't think it's published yet, but looking at using the choroid plexus as sort of a seeding for the analysis. More recently, we've not focused perhaps so much on the choroid plexus itself, but rather the ventricles, the whole thing, because obviously it's going to be soluble, probably mechanisms. So then we'll be looking at this periventricular demyelination patterns and bands of that, also in neuromyelitis patients, which is also related to the ventricles, right, with the reinforcement.
Can maybe add something there, because relating to something that Bob asked, how widely applicable these methodologies are and all that it would be, in my mind, very important to use high resolution imaging and all these new techniques to find molecular correlates that we can then measure, and also in patients over time, potentially with methods that can be applied more universally than only in specific centers.
Yeah, and I mean, neurofilament light has been one of those soluble markers that there's been a lot of hope for. But looking at these studies over time, I mean, they will be very dynamic, so it will not reflect so much the disease course for an individual patient on their trajectory, but rather just how much inflammation or rather neuroaxonal injury there is at a certain time point.
But I think it's very naive to think that one molecule is going to catch the complexity of human disease. So I think we have to go beyond it. We have to look at everything that we can measure and then do a complex models to see which patterns are correlating with specific pathology.
Definitely. Just one comment on your age thing, which is quite interesting conceptually, because now I've been showing you volumetrics and volumes of different tissues, but saying that the thalami are like 17 milliliters doesn't really say much, right? So what's been very popular right now is actually training AI models to look a lot on healthy brain MRIs, and then learning how does a 17-year-old brain look or a 43-year-old brain, and then applying that to MS and look how old does this brain look. And it's been somewhat disputed with it's called brain age estimation, but it's a little bit disputable because it's not normal aging, right? But maybe part of it is. So it would be very interesting to look at this age patterns versus brain age patterns on MRI. That's something we can do tomorrow.
Yeah.
The question that I have is, I mean, we completed in this presentation of parasites accumulating lesions and macrophages, and with your techniques, you see myelin, but will we arrive to a stage that we'll be able to see different cells that are, and that because I mean now we start to know a lot about the biology of the parasites, like Anna was also showing different cell components, but what you are imaging is still not at that level. Will we reach that level, you think?
Yeah, yeah, there are. Can I get some? I mean, to some extent, we're already there. So with PET, you can have ligands of specific cells. So, for example, it's very popular to do TSPO receptor ligand imaging, and then you can look at microglial populations, astrocytes, etc., and definitely for astrocytes as well. Okay, now I'll stop talking.
So perhaps we can form the parasites.
Yeah, I mean, for us, it's more like, of course, I mean, I totally agree. There's a certain complexity, but also, I mean, for me, more like to understand complexity. One needs to simplify it in a way one can mechanistically pinpoint it. So that's why we are trying, and then we're very interested in the translation aspect of it. So that's why I'm very now like also investigating to everything we do, like we're trying to get some human tissue samples that we can analyze to see how similar the processes are. But of course, I mean, we cannot do experiments. We can, that's very much just the characterization of tissue and phenomena that we can validate or see the differences. And I mean, one thing that we're doing, for example, in spinal injury is to establish timelines. So let's say now we'll do the timelines in mice, and then let's say we find maybe coming up in the future with intervention strategy. So in parallel, we're now running timelines to see like how's fibrotic scarring happening in humans to the same kind of cause, because this will be like very important to know exactly when do we need to go in intervene. And these things we can, I mean, that we can do on patient material, maybe even like with imaging techniques, one can, I mean, that's basically what you're saying. I mean, that's early we can diagnose it, but better we have. And I think that's done, needs to be done in correlation with animal models where you may not exactly match the time. I think, you know, certain things take longer in humans than, I mean, you cannot even model aging in animals as it happens in humans. But if you find ways to basically categorize phenomena that you've observed biologically, and then find the parallels in humans, in that way you may be able to you know, go for the same critical points that you want to intervene and interfere with.
Any questions from the audience? Bob?
I was just been sitting here thinking and considering that the title of the workshop was demyelination and remyelination. And we haven't heard too much about remyelination. And I think that's characteristic of the research field. It seems to me that most of the activity is done in the demyelination and very little in the remyelination. So I wonder what your thoughts are. Why do we do that? Is it easier to study demyelination? Are we lazy? Or is it where we should be putting our efforts?
I think the problem is the finding the drugs to treat with, but that actually there are examples in MS with a couple of different compounds that have shown at least some promising effects, but the clinical effects are not as so good, so we you know, could really foresee larger trials, and I don't think we'll see those drugs in the clinic. But the fact that we can change something is promising, and what we need more is more biology and understanding what candidate drugs we could use.
I can maybe add, even though it was not in the title of my talk, so I didn't focus much on it, but when it comes to, for example, autophagy or phagocytic ability of microglia, that's the remyelination mechanism that we're looking at, because we need to remove debris to allow for remyelination. And we're going to start looking into compounds that can potentially boost that process. So I think it's sometimes in the way how we word things, not necessarily what we exactly study.
Yeah, I think it's the same. I mean, there are also already evidence that, I mean, that's the whole purpose if we study scarring and multiple sclerosis, that maybe, you know, blocks also efficient remyelination. So if you find a way to reduce scarring, and there are already like some experimental evidence that if you remove fibrotic scarring, you get at least more oligodendrocytes in the lesion site. So this, I mean, that's the purpose, let's say.
I can say the same thing about the work I presented. In the end, what we are looking for is like a way to change the environment into more like pro-myelinating environment. So, and of course, we know that it's very difficult to access the brain, but if you can access it from the periphery and kind of make it, you know, in this case, correct, like to secrete what we want, and maybe we can kind of promote this pro-remyelination environment for the brain.
And I think I would just add that, I mean, we still have to learn a lot about remyelination in humans. There has been a lot of work in mouse, that given us a lot of insights, but we are still scratching the surface in human research. I mean, we from the mouse work, we assume that OPCs are the main contributor for remyelination, and I think they will have an important component, but also there is studies that suggest that perhaps natural oligodendrocytes also might have a contribution for this, and we still need to learn more to see how can we really promote remyelination.
And maybe just to add to that, also consider the mechanisms of how the neurons fight and survive in the inflammatory environment, even if we don't necessarily have a demyelination connected to that. There would be influences of different soluble mediators, probably also far away. So we have to think about the neurons too.
Yes.
But I'm thinking that if you know which button to press to really kickstart the remyelination, do you envisage that you need the demyelination process to stop first before that can happen? Or do you think that one could end up with a situation where we have competition between those processes? So is the demyelination train running too fast so you can't stop it?
So the question was whether we need to stop the demyelination first before getting into remyelination. I think here we have a couple of different aspects. So we have this inflammation-driven demyelination causing focal lesions, and that we can definitely stop. But then there's the progressive aging demyelination. That's a totally different thing, and that we can't stop. A big challenge is how long time do we need to run a trial to really detect remyelination with imaging? We don't know that. 1 year might be far too short. Maybe it has to be like 3 to 5 years, and that's difficult. So then we need the soluble biomarkers to get some indication to bring an effective treatment to the clinic, then it's costly, it takes a long time, and we need to make more intelligent designs of these studies, the early studies, so we can screen more substances and do it in an efficient way. That's a big challenge.
And also what you mentioned with aging, I think one needs to consider whether there is a certain window of opportunity to act with different types of interventions, both pharmaceutical, but also potentially others. And we know very little about that.
Yes, there's a few papers. The Cambridge group that has done, Alistair Coles and his team has done nice studies, and they have some indications that age is an important thing to keep in mind.
The question there, is that taken into consideration with clinical trials?
Yeah, you must take that into consideration. I mean, you could, of course, restrict your age span for your study, but then that will have implications for what, type of, you can't extrapolate and so on. So I think we need to look at we can't just do studies in 18-year-olds. We need to do, but perhaps looking at, as we do with progressive trials, looking at older patients might not be the best category. So finding imaging markers of the progressive pathology early, before the clinical progression makes itself visible. I think that's important also. We see that from the Alzheimer's field, that you go early, stratifying patients on biomarker profiles. And I think we need to implement that also in MS, because the clinical phenotype comes so late.
So if I can add some, I think maybe to speed up things, I think we should communicate more with the clinicians, with actual medical doctors, because I think that they are seeing a lot of more things that we don't know, and questions. Just now you said that you had some ideas about seeing our work. I'm not sure if we are so good in that. I think we should do it more often, because it could speed up the process and sometimes go to to what it really matters.
Do you think, when aging is such an imminent thing, there's a lot of happening now in terms of senolytic drugs. you think it foreseeable in the future that you have a combination of senolytic drugs and more disease specific, just to in one way fight the aging aspect?
There's one effective treatment already known. It's physical activity, regardless if you have it diagnosed or not. But yes, I do think for progressive MS, as with basically all neurodegenerative diseases, that general aging aspects will cloud everything. So it's difficult to know what's general aging and, let's say, biological aging, or what is the disease-specific mechanism, and also how the disease affects your behavior. Maybe you move much less and you interact with fewer people, and then you get worsen.
So it's not the conclusion, we all need to run more.
Yes.
And talking about running, we have already gone 15 minutes.
Yes.
Half the time.
I would have a few last acknowledgement for all of you. So of course, thank to the speakers for these 4 amazing presentation and also the discussion that them led to. I have to acknowledge, of course, our hosts that I have had the opportunity to introduce. So it's Professor Gonzalo Castelo Branco. He's a professor here at Karolinska in glial cell biology, and he's studying molecular mechanism determining transcriptomics and epigenomic states of oligodendrocyte lineage cells. And then Professor Fredrik Pihl, he is professor of neurology here at Karolinska, and he's the head of research at the Academic Specialist Center here in Stockholm, which is Sweden's largest clinic for multiple sclerosis. So thank you all. Thank you all attending online and here in person. I think it was an amazing discussion and really interesting perspective for the future.
Yes.
We thank also Dr. Prata for the opportunity to organize this discussion between clinicians and also basic scientists. Also Strat Neuro, which is the strategic area for neuroscience here at Karolinska Institute, that also made it possible this to happen. Tony Jimenez Baristine, who was fundamental for all this contribution from Karolinska to happen for this initiative. Also Chiara, yourself, because I think you have been also been very, very important for this. Fredrik, do I miss any more things?
I think
on the speakers.
Yes.
Contributing so much for the also for this lively discussion.
One more, our patients.
And our
patients. We need to interact more also with our patients.
Okay, and with that, I think we conclude the session and give a round of applause for you, the audience, and also for the speakers.
Yeah.
And now